connected? Yes. Okay, just a second. Okay. Okay, wake up. Okay. Thanks much for inviting. It's a real pleasure. First time here, and I really enjoy. I enjoy the views, I enjoy the culture, and surely enjoy the great talks so far. I'm going to talk about actually two things. I'm going to do, I hope, much of the talk going to be about this topic, and then I hope to leave a few, maybe a few minutes at the end to talk about something else. So we're going to start with antibiotic resistance, how we might be able to predict antibiotic resistance, maybe to even think about way we can circumvent antibiotic resistance. And at the end, if there's time, I want to kind of do a big gear shift and talk about a tool, a computational tool that we've developed for interactive data analysis that I think might also be of interest to the audience here. Okay. So we know that antibiotic resistance is growing as a major public health concern. Bacteria have found a way to evade essentially every single antibiotic that has been introduced clinically. And we also see bugs that are becoming resistant to multiple antibiotics. And really, we and others are trying to think about ways we might be able to circumvent that problem, to rethink the basic way we diagnose and treat microbial infections. So for diagnostic, we want to think about anticipatory diagnostic that can tell us not only what the pathogen is capable of doing today, what it might evolve to become tomorrow, and we want to think about that diagnostic as driven by the genome of the pathogen and the electronic health record of the patient. And for treatment, we want to think about new treatments, combinations of antibiotics that maybe has a potential to slow down, perhaps even reverse and drive evolution backwards. For today, for the scope of an AI conference, I'm really only going to focus on the first part. We cannot see. Only going to focus on the first part. I do encourage you to look at this paper we just published on how we might be able to use drugs in combination, but I'm not going to cover this today. Okay, so diagnostic. Here is your problem statement. We have a patient, they come to the physician, and they have an infection. The physician needs to decide what to do. Essentially, if this is in a community, and the physician, need to prescribe an antibiotic, to choose an antibiotic to treat. Okay, and there are two ways she can do that. She has a green path and the red path. The green path is kind of slowly by the book. Let's take a sample, send it to the clinical lab, culture, test what the pathogen is sensitive or resistant to, back after a few days to the physician, and then she can prescribe an antibiotic that perfectly match the susceptibility of the pathogen. Okay, that's option one. Option two, which is also okay by clinical standard, is what we call empirical treatment, in which the physician just skip the long process and just prescribe an antibiotic and hope that it works fine. So we thought, can we come at that point with a machine learning algorithm that can help make those decisions on which antibiotic to prescribe? Well, based on what? Based on two things. Well, I would say, based on, we can imagine going based on the pathogen, and we can imagine going based on the electronic health record of the patient. I would say we've done a lot of work on pathogen genomics, and I only want to show one slide on that. For today, I'm going to focus on the electronic health records. But just to say, for the pathogen genome itself really contains amazing information, which again, not a part of the talk for today, but which you can mine and really reveal from the pathogen genome, and especially how they change in time. You can reveal what are the challenges, the main challenges, physiological challenges that the pathogen experience within our body during infection. At a population level, at a single patient level, and even down to single organ and even single site within these organs. What come out of this type of analysis is response to metabolites, to environmental stress, response to stress of the immune system, and very importantly, response to the treatment stress to the antibiotics. But again, this is not so much the focus for today. For today, we want to focus on how we might be able to prescribe an antibiotic based on the electronic health records of the patient. Specifically, what's available for us is some demographic of the patient, and very importantly, the clinical record, the history of past infection and past use of antibiotics at the individual patient level. Okay. We specifically chose to focus on urinary tract infections as the first model. They are just extremely common, especially for women. There are multiple different drugs to choose from, yet despite treatment, infection often recurs in chronic cycles, infection treatment and recurrence. And finally, there is much growing evidence from our lab, from other labs, that these infections are very commonly seeded by the patient's own microbiome. So we thought that might play a role, that might play kind of a predictive role that we might be able to utilize. Okay. So at that point, we allianced with Maccabi Healthcare Services, back then with Professor Varda Shalev and Professor Gabi Hudik, and now continuing with Sivan Gazit and Tal Patalon. I want to say amazing collaboration, very fruitful, very kind of stimulating for me, and really opened my eyes to new questions and new approaches. And together with them, we've assembled this huge data set of infections throughout Israel of about on a potential cohort of about 2 million members, from which we have about 1 million of recorded infections, and importantly, with phenotypic data, resistance and sensitivity to a panel of antibiotics of the infecting pathogen, and data on the patient, demographic data, and drug and antibiotic use data. Okay. Effectively, you can imagine we kind of have, this is a 12-year worth of data. So you can imagine we kind of have a live map of infection in Israel, amount, who is infected, and resistant profiles. Okay. So we can go and feed all of that to a machine learning algorithm. We use different things. I would say, in honesty, at the end, a logistic regression works just fine. A gradient boosted decision tree works slightly better. But essentially, much of it is about how you extract features from this data, and we can talk about it maybe later. But at the end of the day, we have the demographic data, we have the past infection data. So if I'm looking at a given infection, I can feed in what was the resistant profile of past infections and past use of antibiotics on each individual case. And from that, the model makes a prediction, resistant to that antibiotic, sensitive to that antibiotics, and so on. Okay. So we can start looking at those predictions. And yes, it seems like it's working fine. It can separate sensitive to one antibiotic, resistant to another, and so on. But we said, okay, great, separate, it can make calls, it has an AUC of 0.78, but what does it actually mean in practice? And maybe to ask in a different way, does it provide any information that is new to the physicians at the end? Because everyone knows if I gave an antibiotic last week and did it work, I probably need to change the antibiotic. There are many trivial rules. Okay, so to answer that question, we went back to the data. And remember how I told you the physician can choose one of two things? She can take the green path or the red path. Well, sometimes she would do both. She would prescribe an antibiotic and also give you a cup to pee and send to the clinical lab. And then, in retrospect, she might change the prescription. But essentially, we can look at the cases like that in which there is a prescription and at the same time a sample was sent to the clinic and retrospectively make an assessment on how, by a gold standard, by the lab standard, of how well these prescriptions matched the susceptibility of the infecting organism. Okay. And we can do that for the physician prescription and for the recommendations of the algorithm. Okay. Okay, so here is what we see. So let's start with the left. On the left is the risk that if I'm going to the physician and they prescribe an antibiotic, the left bar, that antibiotic is incorrect, is not matching, namely my infection is actually resistant to that antibiotics. Okay? So the rate of mistakenly giving an antibiotic that is a non-matching antibiotic. And we see it's about 9%, okay? And you can compare it to what happened if you just roll a dice or if you permute the prescription between the patients. That's about what you expect. It's about the rate in which the pathogen are resistant to antibiotics. It's about 9%. Okay. But if we use the prescription from the algorithm on the same exact cases, we can see that we can lower the rate of mistakes by a huge factor, clinically speaking, right? Some 40 or 30% less mismatches of the algorithm compared to the physician on the exact same data set. Importantly, when you do something like that, you might say, okay, but it's a bit unfair because the algorithm may always suggest an antibiotic to which there is very little resistance. Okay? That maybe the physicians are not choosing for many reasons, perhaps even for the exact reason that there is not a lot of resistance and we don't want to select for resistance to this antibiotic. For example, fosfomycin, for people that know, very little resistance in the clinic. So to account for that, we also did something different. We did what we call the constrained model. We told the algorithm, okay, please prescribe an antibiotic for each case, but under the constraint that the only thing you're allowed is to take the exact same prescription that were given by the physician, just permute them between the patients, okay? And you can see that still we can much reduce by some 30%, big factor, the risk of prescribing an antibiotic to which the pathogen is resistant. I want to say that model was published 4 years ago. Since then, we've actually worked with Maccabi to deploy it. It has been deployed a few months ago in practice, in the clinic. So now when a patient comes to the doctor type urinary tract infection, a Windows pop-up that says, an algorithm suggests you give that antibiotic and not that antibiotics. And we're now following up on that to see what actually happened in practice. Does it really work the way we expect? Which physician actually followed those recommendations, and so on. Okay. Okay, so I want to say that has been published, like I said, some 2-3 years ago. And for a while, we've been happy and satisfied. But then we kept thinking about it, and we said, okay, I'm matching an antibiotic to the susceptibility of the pathogen. But is that good? Does that actually help the patient recover from the infection? Okay? So now I can set a different goal. I can say, what I really want is that the infection would go away. That's what I really want, okay? I imagine that a good way to do it is to match the antibiotic to the infection, okay? To the susceptibility, okay? But I can actually go back to the data and look at the actual outcome, clinical outcome of, did the infection go away? Did the patient come back with an infection in a very short time? That would be a failure of the treatment, okay? So we are going into the same data again, but changing what we want to predict. What we now want to predict is the risk of treatment failure, the risk that despite treatment, the patient would come back with an infection within a short time. Okay. Now, I want to say, my state of mind, and I think the state of mind that I've tried to broadcast to you guys, and I think the state of mind that leads much of the way we think about antimicrobial treatment is that what I need to prevent infections, recurrence, is to match the antibiotic to susceptibility of the infection. An infection would recur if I fail to do it correctly. So to a very good extent, a big paradigm that drives antimicrobial treatment is the following paradigm. Antibiotic treatment mismatch, namely I give an antibiotic that is wrong, the pathogen is resistant to it, that's a reason for treatment failures. Okay? I want to say that paradigm is so strong in the way we think about antimicrobial infection that it really drives a lot of the research, a lot of the work. There are many different startups and companies whose job is, let's quickly figure out the susceptibility of the pathogen, because then we can match the antibiotic to those sensitivities, and then everything will work, okay? But if you think about it, in principle, there are other ways treatment can fail. Maybe it was correctly matched, and still the treatment failed, okay? And maybe it was correctly matched, and the pathogen gained resistance to the treatment. That's also a possibility. Okay. So we said, let's just look at the data. Let's find all the cases of treatment failure like that, and see which categories they fall into. And when we did that, I personally was very surprised, because really what happened is that the good part of treatment failures are not because we mismatch the antibiotic. Okay? Like I told you, the chance of mismatching, even if you choose an antibiotic at random, is very small. Okay? It's actually most treatment fails despite the fact that the antibiotic correctly matched the pathogen. Okay? And not only that, they fail with this cyan here by gaining, resistance to the treatment. Okay? So a lot of treatment failure is we correctly choose an antibiotic, it's by the book, it's correct, but the pathogen gain resistant and come back. Okay. How does it gain resistant? You can imagine 3 different ways. Maybe it got a new gene, maybe it got a mutation, and maybe the pathogen went away and a new bug came in. Okay, so we wanted to answer that, and for that we went into a clinical collection stage in which we collected recurrent samples. So we have an infection, and I take the bug, I actually collect the clinical sample, treatment, and we wait and see. If the treatment fails, we collect the next sample. And then we can take those 2 bugs and compare them genomically and see what happens. Okay. So here is how it looks. Here are all the cases that were treated with 1 antibiotic with ciprofloxacin. Okay? And that's the phylogeny of these cases. And they're paired. So each one, there is before and after treatment. I'm going to start connecting them for you. Okay? And let's first connect the one that did not gain resistance. Okay, and here they are. That either started resistant and stayed resistant, or started sensitive and stayed stayed sensitive. And I think what I want you to see here is there are mostly those bloop, 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 the small ones. Very few of these different strain ones. Mostly, it's the same bug come back. Okay, I treat, treatment didn't work, the same bug persists. Okay, but if I look at the one that gained resistance, look what happened. Whoops, it's actually always a new bug. Always a new bug comes in that is resistant to the treatment. Okay, different ways to see that. If you look at the genetic distance, you'd see that the cases in which the pathogen gained resistance are always very different genetically. It's a new bug that comes and reinfects. And not only that, these cases do not appear when the patients are not treated. Okay? So the conclusion from here is the antibiotic itself induces, antibiotic treatment induces reinfection with a new strain specifically resistant to the treatment. Okay. Okay, now can we predict that? Can we predict that? Okay, so we thought about it and we said, okay, if a sensitive strain I'm treating, I'm getting a new strain. Well, where is that new strain coming from? Like I said, much of these infections actually seeded from the patient's own microbiome. We're now collecting microbiomes and future infections, so that's a big study we are doing. But for the sake of just starting, we said, okay, if that infection is coming from the microbiome and we want to predict it, let's look at past infections. They probably also came from the microbiome. Okay? And that means that the risk of finding of the treatment failing, gaining resistant would depend on whether the same patient had a resistant infection sometime in the past. Okay. So that's something we can actually test in our data. So again, we can take all the cases that are correctly treated. The pathogen is sensitive to the antibiotic that is being used, okay? But separate them based on whether sometime in the past the patient had an infection resistant to the treatment. And our hypothesis is, if we separate that, despite the fact that both classes are correctly treated, it is those patients that had a resistant infection in the past that are much more likely to experience treatment failure and gain of antibiotic resistance. And that's indeed exactly what we see. And again, those effects, I want to say, they're not only significant, they're huge. If you look at these numbers here, the risk of an infection recurring with gain of resistance is some 4, 5, 6-fold higher if the patient sometime in the past had an infection resistant to the treatment. Again, despite the fact that the current infection is sensitive. Okay. So again, we can now play a similar game to the one I've shown you before and ask, can we actually now design an algorithm that prescribes an antibiotic, okay, but now not just anticipating whether the pathogen is going to be sensitive or resistant to the antibiotic, but actually anticipating whether the treatment itself is going to work, okay? And we can see that, again, we estimate that we can increase the efficacy of the treatment, but this time with respect to actual treatment failure by a very large amount, even by a factor of 2 or better. Okay. So, to conclude, I think we started with thinking about predicting antibiotic resistance, and I've shown you that patient demographics and past infection and past use of antibiotic can really be used to predict antibiotic resistance. But then we kind of switched mind and we said, okay, but do treatment fail because we failed to predict antibiotic resistance? And I've shown you that, in fact, treatment failure is very often associated not with incorrectly matching an antibiotic to the infection, but rather by the pathogen starting sensitive but gaining resistant to the treatment, with antibiotic inducing gain of resistance and treatment failure. And finally, we asked, can that process be predicted? And I've shown you that yes, it can be predicted based on the electronic health record of the patient, and especially based on whether the patient sometime in the past had an infection resistant to the treatment, with the hypothesis that these infections are very commonly seeded by the patient's own microbiome. Moving forward, we imagined that profiling human microbiome together with future infections would give us another handle, an important handle, to play those games and to even kind of think ahead, not just from one infection to the other, but maybe 2 steps, 3 steps forward, and kind of play it like a chess game that we are using reinforcement learning approaches to anticipate not just the greedy, immediate reward, but to think about what a current treatment would do to future success to our capacity to successfully treat future infections. Okay. So I think I mentioned a lot of this is really enabled by a fantastic collaboration with Maccabi. I mentioned the key people involved in my own lab. Multiple people have contributed that are listed here: Idan Yelin, Morta Lucas Isinova, Matthew Stacy, and others. And I think that would conclude the first part of the talk, and let me see if I have time for the next part. I have like 5 minutes? Okay. 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 So here's a new talk. New talk, we're restarting, but I thought it might be of interest to the audience here. This is, in a way, an introduction, very, very quick introduction. We're not going to dive into anything, but it's an introduction to a new tool that we've developed that we just released. It's completely free and available online. You can look it up in the links here. And it's a tool, it's a Python package. It's a package, open source package for what we call inherently interactive data exploration. And I want to show you the motivation and then just show one quick example, and we're going to end there. Okay. So here is your, let me just move things here. Okay, so here is your, in a way, most canonical data analysis pipeline. We start with a lot of data on one side, we go through a series of analysis steps, and then we present something that Shai might want to call insight at the end. Okay. And I want to say, for many people that come to this for the first time, they kind of imagine these as many black boxes that we combine. But for anyone that went through this process 1 or 2 times, understand that there is a lot of decisions we have to make, a lot of choices, and in particular, a lot of parameters that we have to choose when we do something like that. Okay. So for example, if I'm looking at electronic health records, and at the end I want to say drug A is effective, drug B is not effective, right? On the way, I needed to choose a lot of things. What was the age distribution, the exclusion criteria, and some parameters that are maybe seemingly less important. What was the averaging window, what bin size I used, what background I subtracted from images, and whatever. Okay. And at the end, these choices, these parameters, affect our results, right? And in a way, my feeling is a big part of being a data scientist, and I think also the most enjoyable part, is understanding those dependencies, understanding how a given parameter affects your result, being able to change those parameters and see how they affect the results, and really maybe gain new insight and understand the robustness of your data to changes in parameters. Okay. But being able to do that really brings about 4 key challenges that I've listed here. First, traceability. We need to know what depends on what. Second, reproducibility. If I go and change a parameter, how do I do it in a reproducible, well-documented way. Then efficiency, I change the parameter, I want to recalculate. What do I recalculate? Everything, I just press run, or do I have a way to only calculate the downstream effects of these parameters? And finally, and in a way I think most importantly, is interactivity. How do I play with the data in a fun and interactive way? Okay. So the tool that we developed is called Quibbler, and it's really meant to address those 4 key challenges. And again, we won't go into anything. I just want to show you one really quick example, and we can end there. And in one quick example, we cannot cover everything, but we're going to focus on the questions of interactivity. Okay. Okay. I imagine most people in the audience know Python, and if you don't, you can read Python code at least. So here is a very simple code, just that code, okay? It's a very simple code. It does something very simple. It draws a triangle together with the Pythagorean squares next to it. And if you look in this code, it starts with defining 2 parameters, the a and b of the triangle, and then it goes through a series of essentially plot command and some calculation command, and then it calculates the actual areas of the square, which are plotted here, is this text command. Very, very simple code, okay? Now I bring this code just as an example of almost any code that we use that start with defining parameters. In this case, the a and b of the triangle. And like in any code, what we might want to do is go and change those parameters. And we want to see how when we make those changes, that affects our downstream data. Okay. But to make those changes, typically what we have to do is go to the code and make a change here, and then rerun everything and see what happens. Okay. It would have been much nicer if instead maybe we can do things interactively. Ideally, maybe we can go and drag that point or drag that point. That could have been very, very nice, okay? But to do something like that in Python or in any language is typically difficult. We have to start going from a mindset of a data scientist that just calculate and plot, okay, to a mindset of a program developer that knows things like events and listeners and callbacks, and it's a big series of tools that are available in order to implement interactivity, okay? Okay. Now comes Quibbler. In Quibbler, the beautiful thing is you don't need to learn anything new. You write your own normal code, completely normal. There's only one command to learn. It's called iQuib, input quib. Essentially, you go to your input, and you iQuib your input. Okay, let's do that. iQuib. Okay. But beside that, it's a completely normal code. Let me just clear the screen here and run it. Okay. It's the same exact code. It runs exactly the same way. It makes exactly the same plot. You don't need to worry about anything. But it immediately comes to life. It's immediately interactive without us doing anything. So that means that I can now go and look, and I can just drag this point and move it. And you see that everything downstream would get recalculated. Those numbers would get recalculated and plot. And again, I didn't need to explain anything to the computer in order to do that. But not only that, Quibbler actually understands the entire arithmetic relationships and therefore the geometrical relationship. And that means that I can also do something like that. Okay, or like that, or even, oops. Okay. Okay, so again, this is just a quick example, obviously a kind of a toy example, but the tool is very fun and very powerful, and you can very quickly go and do things that are fun and interactive like that, play with data in many different fields of science and data analysis domains. For image analysis, it's really fun. So to write, let's say, a software that go and crop an image and threshold and all the parameter choices that come with doing analysis on any type of data is immediately interactive out of the box. Okay. Also, because things work in parallel, you can very quickly write analysis that work on a lot of data. And when you change something, only the very specific data item slice within your array, within your data, will get recalculated. So things actually work very fast. Okay. So I think we can end here. In the second part of the talk, if I need to summarize, I've shown a new tool. It's called Quibbler. You can look it up on GitHub. You can pip install it if you want. I want to tell you, if you start playing with it, it's really, really fun. You just go analyze your data the way you're used to, and everything will just come to life automatically. Okay, thank you.
Yeah. Okay, awesome. There is actually this has been.
Are you ready?
Yes, just as a typo, I'll fix it.
Take your time.
Awesome. Okay, so last talk for today before we enter discussions and panel is going to be from Mo Lotfulahi from the Sanger Institute. Please.
Yeah, thank you for the invitation, having me here today. So I'm going to talk about a bunch of applications of machine learning and AI in single-cell biology, but like focused on applications in drug discovery, from understanding the disease and potential targets toward predicting the response of the drugs in this field. Yeah. So a very short introduction about single-cell biology and what type of data that we're dealing with is assume that we got some sample from a brain, and then we zoom in into one cell and then we extract different information from that one cell, be it gene expression, chromatin accessibility, protein abundance, or the location of that cell. And now any type of perturbation that will change the state of the cell, be it drug or a disease, can be modeled using by basically having one sample from the healthy and one sample from the disease. But you understand the single cell differences, and that will allow you to do a bunch of like cool applications. So single cell technologies have been out there for a few years now, maybe more than 10 years. And consortia such as Human Cell Atlas and a few parallel efforts, they try to basically make a map of all the cells in our body and per tissue. So basically you have a table of like tissues instead of elements, similar to that, so a similar analogy. And then you have single cell methods to then sample from this different tissues and understand multiple things first. And this has been basically my research in the past few years. First, use this type of data sets to understand the difference between health and disease. And then modeling how we can perturb a system, either healthy or disease, using drugs and basically modeling that kind of changes. And then, as we know, the response to the disease or the response to the treatment has inherent roots in our genetic codes. So basically understanding the sample variability in this case would be the next goal that you can leverage single-cell biology for this. And similar to the table that you see there, I think the field is going toward using computational methods, machine learning methods to build a digital version of each tissue doing these three different applications. And that would be like the vision, combining both data and also the modeling in this aspect. So I'll talk about single-cell atlases a lot in this talk. And when the word atlas is kind of heard, it refers to a huge thing, so a big data source. And I want to have your attention on a problem here that when we're trying to build an atlas of the cell for each tissue, then we're having a problem which is called batch effect. And that batch effect is basically the technical effect between different studies. And when you want to integrate them, it basically hinders naive integration of the data set. So in the first row, you'll see 4 different studies. So it's a low-dimensional representation of the data. Each dot is the cell. And in the first row, you'll see cells colored by studies. So basically the labs that the sample were obtained, and all of them actually come from pancreas cells. And in the bottom row, you'll see similar cells, so basically the same representation, but colored by different cell types. And if you look at these 3 alpha cells, so clusters of alpha cells, while they're biologically similar and then they have the same function, since they come from different labs, they're actually separated and in different parts of this representation. And in order to continue with downstream analysis, we want to go from the left representation to the right representation where we actually corrected for these technical effects. So now all the alpha cells and also similar cells, irrespective of which data set they come from, they're now aligned on the top of each other. So one of the main goals of building these atlases is basically facilitating and automating the analysis of new data sets. And this has similar analogy in other fields, specifically mapping a new genome to a reference genome. So this is being actually like a usual way that every new genome is now mapped to a reference, and that facilitates the analysis of the new genome because the algorithms for reference mapping are actually faster compared to reference assembly, and the type of data that you need for reference mapping is actually easier to obtain and less expensive compared to reference mapping. to the reference building. So the similar analogy could be used here for single-cell data analysis. Once these large cohort of data sets are integrated, so we can divide it into two steps, reference building. That means that for each cell you have multiple modalities, so gene expression, chromatin accessibility, spatial location, or even clinical metadata from each patient to build a reference, and then have another algorithm that maps a new data on the top of this reference, and you can use this to annotate or and analyze the query data sets. There are a bunch of challenges in reference mapping. So as I said, there are batch effect in the data, so you can't just like use an integrated object and then concatenate it with a new object and use that. So you need to correct for the batch effect while preserving the biological variability. The second thing is dealing with distributed references, meaning that a bunch of like these reference atlasing efforts are collaborative projects, so you always need to send your data somewhere to integrate it for you. And so any reference mapping algorithm would be preferably decentralized, meaning that you don't need to send your data, but you rather share the model that the reference was built with, and you only use the query data set to train that model. And of course, computational efficiency and the ease of use, because once these models get bigger, so the reference models, they get bigger, they're harder to, most of these, like the top performing models based on neural networks, so it's actually hard to, again, fine-tune those specifically for bioinformaticians and people who are not that familiar with machine learning. And the last thing is basically making all of these models accessible for everybody. So this was the goal of a project that we started a few years ago to make a optimization process that would be architecture agnostic, meaning that once you have a neural network that was trained to correct the batch effect for a bunch of reference studies. So the backbone of these models are conditional VAE, so conditional generative models, not just VAE, that receive gene expression as input and also the label of each cell, so basically which study the cell belongs to, and they learn a latent representation that they correct for the batch effect or learn an invariant representation of the data. So the idea was get a pre-trained model, push it into a model repository, and then perform transfer learning or lifelong learning across healthy reference to disease or across different species on organs and studies, while constrained to the criteria that I basically mentioned in the previous slide. So once you have the query data set in this specific architecture, you simply freeze the architecture so that the weights in the model, and then you add these specific nodes to the conditional embedding of this model, and you only fine-tune these nodes using the query data set. That keeps the representation of the data same for the reference data set and also correct for the batch effect. And then you can iteratively update this model using query data set and then push it into a model repository. So this was a paper that published last year, and then kind of like started a wave of like basically model sharing using single-cell data sets. And so I just want to show you a bunch of examples that how people are using this and to basically build these reference atlases. So this is an effort by the human lung community that they built a human lung cell atlas with 500,000 cells, and they used this framework then to update it to 2.2 million cells in 2 years. And so now you have a final version of the atlas with 46 data sets, 445 different individuals, and then across multiple samples and also different diseases. What they did with this atlas is basically use it as a resource now that you can query new data set and project it on this new atlas to identify whether you have novel cell states or subpopulation in the query that are not present in the reference, because you only can find those when you contextualize within a bigger data set. So this is what they did. And so the gray one is query, and then gray one is reference, and the colored one is query. And they used a super simple KNN classifier on the latent space of this model to transfer cell type annotation from the reference to the query. And what you see here is basically uncertainty of this model, super simple uncertainty based on the distance. And using this uncertainty, they could identify novel cell states which are in the query and not in the reference. So here you see the distribution of the correct, incorrect, or unknown that they obtained using this uncertainty metric. And indeed, the ones that were incorrectly labeled or unknown, they were actually novel cell states. Another example is from a recent publication that I like is basically using similar notion to first build a single nucleus resolution reference for human prefrontal cortex across development. So starting from very early ages toward like more mature states. And they use this reference to identify the difference or the gene programs that are actually preserved across the development and also continue to disease. So by basically mapping glioblastoma or different developmental stages. And they even use this reference atlas, so in vivo model, to map in vitro data to potentially compare what are the preserved states in the in vitro organoid data from brain that was also in the reference data set. So, and also one basically fun predictive task that they measured, that they designed here was basically predicting a single cell age from the reference to the query, and they showed that using this they can identify and they correct for the correct biological age in this case. So what was interesting here is, and what I find fascinating is you can now look at these reference atlases as a database. So you can query them with new data and ask more high-level questions. Which are gene programs that are changed after disease, or where do disease risk genes act, or what is the effect of a certain drug? The problem with these models are they're all based on highly nonlinear neural networks, so they're not quite interpretable, so you don't know the axis of the variation of the latent space of these models. So the ideal setting is if we can go from non-interpretable setting to more interpretable setting where each axis of variation is now in like a known entity that a biologist or a medical doctor can analyze. So basically T-cell differentiation, inflammation. And that drove us to make these models a bit more interpretable. So what we did was we mined papers, literature, databases, and also got expert data. And we built something that we call gene program matrix, where you see a binary matrix where each row is a gene program and each column is basically a gene, and the 0 means that gene is part of the gene program or is not, and 1 is basically the membership of that gene in that gene program. So the idea was, can we make each latent dimension of this neural network in a way that it captures the variation with respect to those specific gene programs? And what we did in this way was basically we designed a mask using that matrix, and then we made the linear decoder so that we can apply that mask. But we also devised a bunch of strategies to correct for the genes within each gene program, so refine the genes in that gene program using the data, and also filter the gene programs that are basically relevant for each data set. And that was in the reference building part, and then reference mapping part, we allowed the model to learn novel gene programs. So basically, if you have a disease data and you built your model on a healthy data, then we basically add new nodes into in the bottleneck, and then we impose these nodes. Since we actually constructed a information bottleneck, the new disease variation will be captured here, and then we make these new nodes, we enforce them to basically be independent with respect to the previous one to further reduce the redundancy. I just want to show a vignette here that here we built a PBMC reference across 5 studies, and then we mapped we mapped a new query dataset that contained cells that came from interferon-stimulated cells as a cytokine that induces a strong change in gene expression, and also contained some control cells. So as you see here, after the mapping, the control cells were mapped to the controls in the reference, and these interferon cells were separated. Since this model is probabilistic, you can actually perform differential gene program analysis and identify which gene programs, which latent dimension can actually separate between the control and the stimulated. And in this case, it was obvious because interference signaling was there. And we continued this analysis by mapping, and if you visualize the data into these two dimensions, you can see that these are actually the main axes of variation, and you can see the changes in this way. So we use this reference to map patient data from so COVID patient treated with tocilizumab. That's basically an IL-6 inhibitor. And these patients were different diseases, the disease stages, so basically severe COVID and then remission. And we wanted to see which cell types are actually responding to these drugs. So the reference was 5-BMC dataset, and we mapped 2 query dataset, interferon beta, and also COVID-19 patient data across 3 different time points. What we observed was almost all the cells from the query patients, they mapped to the reference, so they integrated with the healthy reference, except 2 cell types. And those were CD8T and also CD14 monocytes. So we wanted to see which gene programs are basically responsive for this phenomena that we see here. And we performed a similar differential gene program analysis, and then we identified signaling pathways, so GPCR, and also that's for CD8T, and also for CD14 monocytes, BRIG1, MDA5, so basically pathogen receptor pathways that basically regulate immune response. And these were basically the ones that differentially expressed in the latent space between this patient data and also the control one. So this gave us a hint that the reason that these cells are not responding to the drugs is basically active cellular communication. And then we followed this up specifically in patient 2, and then we found out indeed like CD8 T cells and CD4 monocytes are basically in communication, and that also changes in the later stages, even when the patient is actually treated. And in the remission stage, that still continues. So that could give us while the treatment with tocilizumab is not, so it has not been reported to be a consistent treatment for COVID-19 patients, but it could give us, so in this case, the differential pathway was annexin-regulated pathways that could give us like certain pathways that might be potentially druggable. And if you find a certain drug for those, then you could potentially you could have prevented cytokine storm, which was the main reason for many of the hospitalization for COVID-19 patients. So in the second part of the talk, I'm going to talk about once we identified those cellular states that are affected after disease, can we actually predict the response of those cells with respect to certain perturbations, be it genetic perturbations or small molecules. So the idea is, since single-cell technology is an expensive type of experiment that you could do, so once you want to generate a perturbation data set, you can't explore all possible drug combinations or potential small molecules. So you start with a sparse set of those. And the idea is, so we had like a series of work on this that whether we can have an in silico model that could predict the missing combinations or missing perturbation that you could have not measured in the initial experiment. And then use those to design a new experiment. So you want to perform a prediction problem that given a control cell, you want to predict the expression of this control cell after a certain perturbation. So I'm going to talk about something that I did 2 years ago when I was at Facebook AI to basically computationally model this problem. So the idea was using interpretable models that a lot of biologists use in day-to-day analysis, or linear models, that decompose gene expression into semantically interpretable components. So the idea was, can we model overall expression as a basal state, meaning that a state of gene expression that we corrected for the effect of the perturbation, cell types, and etc., and then we learn the linear composition, but in a latent space. So the idea was we encode the gene expression, and then we learn a latent representation of the cell, which we call basal state, and then we learn the representation for cell type, perturbation, or even dosages, and then we linearly add them up, and then we decode this linear composition, and then we predict the gene expression. So at the test time, if you measured cell A with 2 drugs, you can potentially correct for those 2 drugs and get the basal state for cell A. Now you've seen cell B in your study, so where you measure cell B with 3 different drugs, and you can perform a counterfactual prediction using these 3 different drugs, and then predict the effect of that cell as if it was treated with 3 different drugs. So we applied this model on a data set. It was one of the first single-cell, highly multiplexed data sets from Cole Trapner lab from 3 cancer cell line, 188 drugs for different dosages. So in total, 4,500 unique conditions and 500,000 cells across 3 different cancer cell line. And then we applied our model on this data set. And what this model gives you is it also gives you a perturbation space, because it actually learns these perturbation embeddings. So for each drug, it basically learns an embedding, and then you can visualize this embedding in a way that, and semantically and also like biologically, the drugs that had similar gene expression response, they actually cluster together. So you could see drugs which had MOE of epigenetic regulation or cell cycle regulation, they're actually clustered together. While for the cell lines, since they are super different cell lines, so we just use this kind of like 2D representation, they're really like not close to each other in this sense. So we tested this model to see whether if we leave out a certain cell, cell type and a specific dosage, whether it can potentially predict the dose response per single cell for the missing dosages of a certain drug. And indeed, it was a sanity check that it will work. And so we actually did this, and then we submitted the paper, and the reviews came back, and then the reviewer said, so what, so you predict stuff, but then what is the usage of this? Can you actually design an experiment with this? And then we used this map that we had before, so that resembles the gene expression similarity. And then we designed a follow-up drug combination experiment using this. So the idea was we selected from different parts of this manifold, and we selected in total 32 mono and then combo drugs, in total 60K cells. And then we fine-tuned this model with this new data set, and we wanted to see whether the model, so this is the new data set, whether the model can now, so first, whether these drug combinations were selected correctly, because they actually mimic, so basically they were selected based on the fact that these are quite different from the control cells because they clustered from the other drugs, and whether the model could predict basically these drug combinations that it hasn't seen. So we divided these different combinations into, so from green to red, from basically easier split to harder splits, and then we showed the model works well. At that point, there was no other competing model, so we kind of like made a super simple linear baseline, and then what you see is basically R2 mean gene expression prediction of control, real cells versus predicted, and also the same value for the differentially expressed genes. And then you see that since there were also a bunch of like synergistic interaction, the linear model potentially failed in this case. So we extended this model into, this is a recent NeurIPS, so basically this year's NeurIPS publication that with the previous architecture, you could not predict the effect of the unseen drugs, and also the other limitation here is so you could not potentially, because there is a lack of data for single cell screening. So the idea was, can we actually use what is out there, so basically bulk data sets to do transfer learning between bulk and single cell. So what we did was we just tweaked this architecture in a way that now we added a chemical prior to this model, so you could just feed in the chemical structure of the molecules that you haven't seen, allow you to predict the unseen effects. And then we devised a transfer learning strategy to transfer from bulk, which is quite different from single cell. And then we showed basically combination of both of these models, so pre-training on bulk and non-pre-training, and then also baseline the pre-trained model performs best and also compensate for the lack of the data. So one last example here is we applied this model not on the small molecules, but also to predict genetic perturbations. So this is a data set from Jonathan Weissman lab in MIT, where they generated a data set with K562 cells, and then they measured 287 single gene and pairs of knockout. And then what the model captured was it grouped the combination of genes that induce similar phenotypes, so basically the pioneer factors or cells that induce erythroid lineages, or the perturbation that like arrest the cell in the cell cycle, they all grouped together. So the idea was then to test whether this model can predict interesting interactions. So in this case, we tested whether the model could predict a synergistic interaction, and also epistatic interaction. So you'll see that like in the epistatic one, the DUSP9 plus ETS, it basically carries the effect of DUSP9, so that masked the ETS effect, and indeed that was basically what the model was also picked up. But what was interesting in this case is we wanted to see whether we can impute all missing combinations in the data. So in this case, they only measured 284 out of 5,329 perturbation combinations. So we, in silico, generated 50 cells for all the missing combination. And what you see here is the mean gene expression of all those 50 cells that we generated. And we also have a uncertainty metric that basically shows you once you go away from like the training data trivially, then the uncertainty increases. That could give you like an insight of like what would be the next set of perturbation that would induce to efficiently explore this perturbation space. And one last thing is similar models could also be applied not on gene expression data, but also perform counterfactual predictions on imaging data, where you have painting, so cell painting data set, and then you want to predict how the morphology of a cell would change if you apply a certain drug. So here's an example of what the model, so it's a generative model, so you give it the image of a control cell, and then you give it a representation of a drug, and it will predict how this cell will respond to this drug. And in this case, it predicted the drug will perturb the nuclear integrity of the cell. And then we applied, we compared this to the state-of-the-art model, which is out there for conditional generation of images conditioned on small molecules. So this was just like two parts of how application of machine learning and single-cell biology could be used to identify disease-related states and also predict the responses to the treatment. And if you're interested in other parts, so I've been lucky enough to involve in like a bunch of projects that combine single-cell biology with population analysis or multimodal integration. We have models that now receive T-cell receptor or like antibodies and dynamics and also building hierarchy. So if you're interested, please reach out. So in conclusion, we can use single-cell biology and machine learning to understand cell type-specific changes in potential targets and then predict the behaviors to those. So this was the work of a lot of people who I was lucky to work with, and so I should mention their names, which, so basically all of these would not be possible without them. Thank you very much.
Okay, thank you for both speakers. And we'll open the floor for some Q&A and discussions.
Hello, doctor. I'm a PhD student here, and my name is Ding. And well, our group is also working on the gene perturbation prediction problem, just like your CPA does. And so I have a question that is, so as you mentioned, the control expressions and all the perturbed expressions are not from the same cells, right? So how to, scientifically decide if these expressions, like control expressions and perturbed expressions, they are independent or dependent? Because in most of the models, we predict perturbed expressions from the control expressions, just like they are one-to-one paired, but they are not actually. So my question is...
Yeah, so you don't have pair data for this. What you need to do is basically then either find out the way as we did to correct for the perturbation effect, which might not be perfect, of course, or you work on the distributional, so basically on the distribution level to see whether you can match the distribution of the data. And there are also a bunch of work after us. They use optimal transport or like different type of like distribution matching or like GANs. To potentially tackle that problem, because single cell technologies are destructive. So there are new experiments that like also technologies that they try to like solve this, but it's very experimental, so it's not out there.
I see, just like a generative model. Just
So you have to go on the distribution level instead of like a single cell level, or find a way to, as we did. But both of these things have their own pros and cons. So yeah.
Okay. Thanks, doctor.
No worries.
Hi. So thanks for a great talk. So sorry, my question is for Roy. So I was wondering, how stable is the distribution? I thought that resistance and the strains change over seasons and over the years. Have you seen that?
Okay, yes, we did look. There is a change over time in general, and there is seasonal change. In the model, we do account, part of the features are seasonality, and part of the features are just calendar date. They contribute, but not a ton. They contribute mainly to the risk of infection, less so for the risk of given an infection, whether it's sensitive or resistant.
And a follow-up on that, so I don't know if it's a strong effect, but do you think if, for example, if the entire community in Israel would adopt your algorithm, do you think the distribution of resistance would change maybe in unpredictable ways and new strains will emerge? This relates to your research.
Right, so that's a beautiful question. I think in general, when you treat microbial infection very differently from when you treat almost a non-infectious disease, you are doing two things at the same time. You're treating the patient and you're affecting the population of pathogens and therefore future infections for the community. With antibiotics, it's a big challenge, and sometimes you, in a way, there's a trade-off between the two things. I think the most obvious trade-off is you can always choose a drug for which there is very little risk of resistance today, but we don't want to do it because we don't want to increase the risk for tomorrow. Whether actually following the algorithmic suggestion would make an effect, and what would be that effect? I want to say first, of course, we don't know for sure, but I want to say just the principle of correctly matching an antibiotic to an infection is overall a good thing, because that minimizes the overall use of antibiotics. Because when you don't correctly match, you follow up with another antibiotic that then match. And when you didn't correctly match, it's not like the antibiotic didn't do anything, but it actually did is selecting for other bugs in your microbiome to become resistant. So that's overall, it's wrong use is a misuse of antibiotics. So I think the algorithm has some potential by correctly matching to susceptibility, and more so the new algorithm correctly matching to reduce treatment failure and recurrent infections. Both of these have the potential to reduce the overall use of antibiotics. So from that point of view, I would expect they would act in favor of reducing, or at least not increasing, the overall resistance in the population. I also want to say, maybe connecting your two questions, when we look at graphs that are published kind of for the general public, that let's say if you look at articles in the New York Times, you see antibiotic resistant rate in the population going up. In practice, it's not so much like that. If you look at a decade-long data set that we are now looking at, The same, maybe go up a bit, maybe for some other antibiotic goes down a bit. Mostly it's quite stable actually. The risk of an infection being resistant to a given drug, quite stable. And what actually creates that stability is a big, big puzzle, I think, for me. Because evolutionarily speaking, if there is an advantage to becoming to a resistant pathogen, it should take over, and if there is a disadvantage, it should disappear. But it kind of hovers there, and it kind of suggests that there are some balancing forces that we don't understand, and maybe they are behavioral, maybe the use of drugs. I mean, it is a negative feedback, but maybe some other things. Okay, so I think overall a complicated answer to a good question. Yes.
Also, thank you for the great
Thanks.
presentations. Also, a question for Roy. So you didn't go into details of how the allocation of the recommendation was done. So what was the model behind the scenes of trying to create the best prediction of which antibiotics to use? But you did show that it reduces or potentially should reduce the resistance. So I was wondering, do you see it more of a prediction problem or a causal problem? Because I think behind the scenes, like a gray area of whether you would try to predict the rate of or the chance of resistance for each antibiotic in a different prediction model for each type of antibiotics, and then, like, I don't know, choose the one with the least probability of resistance, or whether you would go about it as a proper causal question of trying to map all the potential confoundings of choosing between drug A, B, and C, and going through the treatment assignment mechanism behind the scenes of why the physician chose that prescription, and maybe different physicians like different antibiotics, so it's like an instrumental variable. So how did you go about this analysis? More of a predictive problem or a causal problem?
Okay, a very good question, which I didn't touch at all, but in a way that goes to the heart of what we actually did there, because I think you're completely right that we are developing a predictive model for each antibiotic. And I have a model to predict resistance sensitive to antibiotic A and to antibiotic B, a different model, and to antibiotic C, another model. But at the end, when I come to prescribe a drug, I need to choose one. Okay, and how do I do that given the output of these different models it's a challenge. Taking the one for which the predicted risk of resistance is the smallest, well, it's a possibility, but it doesn't account for many things. As some of those we've actually raised, like the desire not to give drugs for which is not much resistant, maybe cost of the drugs. There are multiple different factors, side effects, multiple different factors that affect to what extent a given drug is actually prescribed. So, therefore, what we actually did is what we call the constrained model, which said we minimize the risk of overall risk of prescribing a non-matched antibiotic, minimize that risk over the entire population, given the constraint that the algorithm is only allowed to use the drugs at the rate prescribed by the physician, just permute them between the patients. Okay. How to do that? It's not very difficult, but it's mathematically totally solvable. And in a way, I think the most emails that I'm getting about this paper is, can you send us that algorithm? It's actually fairly simple. We can talk about it later. And I think in a way, it's something very general that appear when you have models that, like you said, individual model for different possible course of actions, and at the end you need to choose one. How to do that is, I think, something that is, again, it's mathematically precisely solvable, and I think maybe is more general than this specific application. Yes.
Okay now? Cool. Great talks. Question for Mo. So, if I understand correctly, the embedding model is based on training data that is based on many manual annotations. Is that correct?
And which part? The first part, or the drop response prediction?
The first part, I think.
So the embedding model is unsupervised, so it doesn't have access to the cell type labels. So it's completely unsupervised. It's basically a generative model of the count data that you have.
I see.
So all the cell type and supervised parts were basically later. I mean, we have semi-supervised models that also use the cell type labels, but that was not what I showed, yeah.
Okay, so maybe I got it wrong, I guess. Is there a part where you do use manually curated annotations?
Yes. So we have like, so in the last slide, we have a model that actually leverages those, but that it could actually bias the representation because these depending on how you infer these like cell type labels, you could force some cell types that you don't want to integrate if you had error in your annotation to kind of like be aligned.
Exactly. So that's exactly what I was
wondering.
wondering
So it's a good bias. So most of the time, actually, the best performing models are the semi-supervised models. But in like the usual practice is you don't use it. So you first integrate the data, and then based on this, you do de novo clustering. And to build the first atlas, I mean, if you have like a comprehensive reference atlas, which is well annotated, then you can just trust it, yeah.
Got it. But then how do you apply the annotations on the clusters that were formed? Is that, again, based on manual
so that's basically, that's
effort?
a tedious and time-consuming unsupervised clustering. Talk to the expert, identify the marker genes, back and forth, sub-clustering. Takes some time, yeah. It's not a fun thing to do.
Cool. AI can solve everything. Thanks.
I just had a brief question for Roy then. So I was really impressed that your system has been taken up and used in the Maccabi healthcare system. And then just following on the questions we had, might there be any opportunities then to, I don't know, run a prospective trial of some treatment options so then you can do I guess a randomization that we can learn from?
Right. First, I want to say it is. I grew up much of my career in kind of like more classical science, experimental science, and even modeling, but all within the context of a lab. And I think the idea of actually developing something that gets deployed in the clinic is there's something very satisfying getting there. And much to be said about the prospect of that is really a very close collaboration like the one we have. Surely that ability opens up new direction for follow-up research on the type of, I think with several different questions on to what extent the algorithm actually succeed in the clinic, in real life compared to what we expect. What does it do to overall to the level of the community? And also some questions about there's a lot that can be asked about the clinical practice itself. And these questions, in a way, to me, they're also fascinating, like to what extent physicians choose to follow recommendations, which physicians choose to follow recommendations, what do you need to do, how to present the data in a way that will cause physicians to follow recommendation. Is it just, hey, I suggest drug A, or what to actually give the reasoning? I think there are many questions that open up in that type of analysis that are maybe they are not in the world of the pathogen so much, or more in the psychological worlds of patients, of physicians. But I think they're in a way very general to how we think about AI may actually interact at the end with the actual clinical system, and they are not less fascinating.
Yeah, and just to follow up with a brief comment, I think or a potential next step for AI then is prospective trials of or of AI technologies, where we can do it, of course.
By the way, even before we think about the algorithm, remember how I said we can go and analyze the data to retrospectively look at cases in which the physician did two things. They gave you a cup to pee and to get the actual resistant profile of the pathogen and prescribe an antibiotic, right? And then on these cases, retrospectively, we can actually say wrong or right, correct or not. Okay. And I gave you, I said, overall 9% wrong. You can start going just on that, no algorithm at all, just on that, and start asking, is that different between young and more experienced physicians? Is that different between, I don't know, experts? Is that different in the more rural areas of Israel or Tel Aviv? There are many questions like that to ask, because it's a place where we actually have an in situ gold standard to say correct or wrong, independent, and I think it just opens up, again, a set of questions that are not in the world of AI per se, but they are in the world of data science applied to the clinics that are quite fascinating. Yeah.
More questions? Yeah.
Thank you for the interesting talk. I have a question to Dr. Mo. In your studies, did you manage to find which the principles that allow us to find synergistic combination? Which principles, especially in biological terms, that allow for a combination to be synergistic?
Well, I think there are two aspects to that. Whether you can actually experimentally can measure that, so you can have a viability assay and then check the number of cells that survive. So basically have a design experiment like that, and then have a simple linear model on the top of that that could potentially, just a super simple linear model that shows that A plus B is, so you can have a linear model that you have drug A, drug B, and then like noise. And then when the nonlinearity actually, or the noise part actually increases, then that shows that AB would be stronger, like bigger than A plus B. So we actually use this super simple metric like that to then assign, based on the weights that we learn for the linear model, and then to assign functions, so whether something is synergistic or epistatic. And that's a bit of also dependent on how you interpret things, and then the parameters so you have to have a cutoff. Okay, at this threshold I call things linear, at additive at some point I call them this and that. So having a model and then after that some sort of thresholding, yeah, that's how we also did it. But yeah, there are fancier models that they also predict the synergy, but as a supervised, but then you need to have labels for that, yeah.
Yeah, because there is, for example, database Synergy H.
Yeah.
Yeah, and it would be very interesting to have an algorithm that could really predict
those type
synergy.
Of stuff, so that database and also the other databases were not measured at single cell level, yeah. So yeah, they're at with the different assays. So you could use that, but I think for this type of data set it would be hard to kind of incorporate those. Mm-hmm. Yeah, because it's kind of like orange apple type of stuff. So.
Yeah, but actually all these databases, they do not contain synergistic effects, only additive ones. And if, for example, we repeat these experiments, for example, having additive successful combinations and using your model, will we be able to predict synergistic ones?
Oh, yeah, that's what we showed. Yeah, that's what we showed. But the concept of finding a new synergy, it's like finding a needle in a haystack, because it depends on the dose, and then a lot of things should go right in that order to find that. But with this model, we actually show that. So if there are synergistic interactions for the drugs that had similar interaction with other drugs, then you're able to predict those. But if it's like it's a super different interaction, like a neomorphic interaction, you've never seen it, then these models basically fail.
Okay,
Yeah.
thank you.
No worries.
Thank you very much for the presentations. I have a question for you, Dr. Mo.
Yeah.
So it's clear that in your research there is a central point, which is how to design a latent space,
Yeah.
which is convenient to do or to make perturbations of your cells, right?
Yeah.
So did you try to see what is the secret in your network or in your model to better model the original data? That's one question. What are the advantages of these kind of variational autoencoders over other models like GANs or autodecoders? And yeah, let's start with these two questions.
So I showed two classes of models. So the first class of models, they were actually VAE type of models, and the second class were basically adversarial type of training, because we use adversarial classifiers to correct for that. So we used and designed both of these types of models. I think the key is these models would not work out of the box, so you need to adapt to the distribution of the data. You need to put a lot of prior knowledge. That's what I also showed in different parts, either chemical structure or like gene programs. And indeed, in the case of the gene programs, even though we reduced the capacity of the model to the nonlinear encoder and linear decoder, we showed that in this class of models, if you optimize, then you have a lower variational gap for the variational inference, because the model, it's easier for it to find a better solution compared to nonlinear models. And if you remove the prior knowledge, then it basically goes back to the similar class of models. So it was also fascinating for us that like to see how much information, how much like performance boost you would get by adding simple prior knowledge into the model, yeah, compared to the model complexity.
Yeah, at some stage you showed a very interesting example how to perturbate using a chemical drug.
Yeah.
So how real is the prediction?
Oh, that's a good question, I think. So we had like multiple metrics. So a gene level metric to see how good you predict the upregulation or downregulation based on log-fold changes. So those are like gene-specific ones. We also had distributional measures to check how, on like a distributional measure, you map two different populations to each other. So, and obviously compared to the baseline methods, we were better in that case. But it also depends on the type of the data and then how well that gene is expressed, the mean gene expression. So these models basically tend to fail for the genes that are lowly expressed. And so this data is not the cleanest data set that you play with, because it has a lot of zeros. And then especially in the cases of like the perturb-seq data sets, they're also super sparse. So the number of reads that you get per gene is also not super good. So if you zoom in into some genes, then you can potentially see the failures, especially for the ones that are lowly expressed. Yeah.
Maybe a last one. Do you see any immediate informations other than the genome data that we can use to better model these perturbations? I mean, for example, the morphology, the dynamics of the cells.
Oh yeah, yeah. So we not in the case of perturbation, what we use dynamics. So basically, spliced and unspliced reads that you get from these experiments. We have one paper that basically, in collaboration with Nir Yosef, that we model like we predict the RNA velocity. So basically, velocity of a cell that goes toward the specific lineage or not. Yeah.
Thank you.
Okay. Just maybe I want to ask a final question to Rui. So first, a clarification. In those cases where you showed or you think that there's antibiotics is given, but it's not because of acquired resistance, but it fails to treat the infection, are those cases where basically you think there were multiple strains residing in the patient and just the antibiotics then selected the other one? That's what you think, or? And related to that, maybe you can answer both, is how much evidence do we have for where these strains are actually coming from? You mentioned the microbiome, but do we know it's coming from the microbiome or elsewhere?
let's start with the second one. On the second one, do infections come from the microbiome? Yes, there are some works in the literature showing that good fraction depending on exact indication. And first, this is for UTI, so other infection may be less. Although wound probably also, but skin microbiome. We are doing now fairly systematic collections of microbiome and future infections, and it's very common. It's more than 50% of the infections we can find exact, like complete coverage, strain isolated from the microbiome.
You're collecting gut microbiome.
Gut microbiome.
Yeah.
Gut microbiome for UTI in women. Right. The other question was, is it possible that the resistant strain that came up was already residing? Yeah, it's certainly possible. There are multiple levels of residing. It can be residing in the microbiome. It can be residing in the urine at low quantities that were not originally detected, and it can be somewhere in between. For example, maybe it's in kind of, okay, there's bacteria, mostly E. coli, they can go intracellular, so they can go in the epithelial cells of the bladder, so that's also a possibility. In a way, part of me really want to know, another part of me relates to your answer to the question I asked. Sometimes we can make those predictions and get a lot of understanding
Okay.
even without knowing. So.
You weren't happy with my answer, but no, okay. That's fine.
Yes.
Okay.
Okay, thanks.
Thanks. So on that note, let's thank the two speakers again for wonderful presentations. We're going to take a break and be back at 4 for a panel discussion of all 6 speakers of the day.